This journey of the developing fetal heart begins in the middle of the 3rd week of embryological development. After the process of gastrulation, the embryonic disc consists of 3 layers called endoderm, mesoderm, and ectoderm. All 3 derived from the epiblast layer of the bilaminar germ disc. These 3 embryonic layers eventually make contact with the extraembryonic endoderm, mesoderm, and ectoderm lining the yolk sac and amniotic sac. During the embryonic period, it is the mesoderm, specifically lateral plate mesoderm, which gives rise to majority of the cardiovascular system. The process of heart development takes place in the previous mentioned embryonic mesoderm, which lies deep to the ectoderm. Progenitor heart cells migrate from the epiblast through the primitive streak into the visceral layer of lateral plate mesoderm towards the cranial end, forming the U-shaped primary heart field. These progenitor heart cells are stimulated by the endoderm to form blood islands. These unite from either side of the embryo to create a horseshoe-shaped tube at the cranial end within the lateral plate mesoderm, essentially forming the cardiac region at the head end of the embryo. The embryo folds cephalocaudally and laterally from day 18 to day 22. The process of lateral folding helps the paired endocardial tubes to fuse caudally except at their caudalmost end. This forms a single endocardial tube or cardiac tube. Cephalocaudal folding helps to position the cardiac tube, which was developing at the head of the embryo, into the thorax. Now, with the transverse section at this level, we will visualize lateral folding of the embryo, a process which aids in the formation of the cardiac tube. Here we are looking at the cross-sectional view for the cranial end of the embryo. The mesoderm has 3 parts: paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. We are concerned with lateral plate mesoderm when learning about cardiovascular development. It is divided into the dorsal part called somatopleuric or parietal mesoderm and ventral part called the splanchnopluric or visceral mesoderm. The paired endocardial tubes and dorsal aortae develop within the splanchnopluric mesoderm. The process of lateral folding of the embryo begins from day 18 of embryonic development and ends around day 22 with fusion of the ventral body wall in the midline. During lateral folding, the mesoderm comes closer to midline for fusion and the endoderm starts to invaginate posteriorly. This process brings the paired cardiac tubes in close vicinity, ready to be fused caudally except at the caudalmost end. The splanchnopluric mesoderm along with paired endocardial tubes fuses in the midline. So does the somatopleuric mesoderm. The space between these 2 layers of lateral plate mesoderm is known as intraembryonic cavity. After lateral folding, this space forms the pericardial cavity surrounding the cardiac tube. Lateral folding of the embryo is completed by day 22 and results in a single endocardial tube within the pericardial cavity. The tube is initially attached to the dorsal side of pericardial cavity by a fold of mesodermal tissue, the dorsal mesocardium. Here we can visualize the ectoderm covering the entire embryo, formation of the neural tube, endocardial tube, dorsal aortae, and the endoderm lining the gut tube. Lateral folding also leads to separation of the extraembryonic layers and the embryo becomes suspended within the amniotic sac, a fluid-filled cavity represented as a transparent layer here. The journey of the fetal heart begins as a tube, which soon starts to undergo the process of cardiac looping and further structural development to resemble the adult heart.